Hey everybody, it's Mr. Fabek here. It's been a while, but now that we're starting to get into genetics, it's probably important to start talking about babies and how babies are created. So there's tons of movies out there. Junior, Knocked Up, Gattaca, Juno. These are great films. Kind of interesting, kind of funny, kind of thinking about how babies are going to be created in the future. It's great stuff. So if you get a chance to ever watch some of these films, you should. And let's do it. Let's talk about how babies are made. So here we go. Objectives. By the end of today, you're going to be able to use these terms like fertilization, embryo, zygote, stem cells, a whole bunch of other ones that go along with the whole process of this making babies or baby science. And then you're going to be able to explain what happened after fertilization and be able to explain pros and cons of stem cells. So you'll know you mastered this if you be able to explain how babies are made, essentially. And we're going to talk about a controversy on what happens when we use some of those materials of developing babies. So let's see it. Here we go. First thing we have to do is really talk about meiosis. Meiosis sounds like mitosis, which we remember is where we have our chromosomes in the middle, and then we have our anaphase, telophase, all of that, right? But meiosis is the whole process of making sperm and egg cells, or making gametes, okay? So the big thing about meiosis is as you are replicating your DNA, replicating your chromosomes, you eventually separate those chromosomes into separate cells, and then you divide again. So essentially you're dividing twice. You're going through the stages of meiosis twice, which gives you different numbers of chromosomes. So what we call a normal cell, like in mitosis, a normal cell in mitosis is called diploid, whereas a sex cell, like sperm or an egg, those are called haploid cells because they have half the DNA. Okay, so diploid is full DNA, haploid is half DNA, and we're only talking about sperm and egg cells when we're talking about haploid cells. So why is this important? Really think about why would it be important that you only give half of your DNA? So what you really want to think about is that when you need to fuse your DNA with someone else's, right, sperm and egg, they have to have the correct number of chromosomes, and if they don't, that's where we can start to see genetic disorders. Okay? So if they have too many chromosomes or too less, we're going to see problems. Also, the cool thing about this whole idea of meiosis is this is where we get genetic diversity. This is where you don't look exactly like your parents or your siblings. You really can look very similar, but it's this separation of chromosomes and then this fusion of new combinations that really give us this genetic diversity. So if you're an animal out there in the wild, this is how we have changes over time happening, this natural selection, which we'll talk about later on. But this diversity can really help species thrive and really creates a fun, interesting planet. So, how are babies made? From a stork, right? But what we really know is you need two people, right? You need a sperm cell and an egg cell. And those sperm and egg cells are called gametes. Gametes. Gametes are a sex cell, okay? So once you have your gametes and you combine them in some way, you're going to have what's called a diploid zygote. Remember, diploid's a full set of chromosomes. So we can see here we went from N, which means haploid. You only have 23 chromosomes in humans. 23 from mom, 23 from dad, for a total of 46. And so this fertilization, and we have the whole process of when they're just uniting and making a nucleus. And then we start to make our zygote, which is the first part, okay? So you guys were all once little baby zygotes. And that's where we have the sperm and egg cells and the chromosomes in the nucleus. And we're starting to divide. And we get this, a bunch of new cells through mitosis, and we'll eventually get into this whole differentiation in just a minute. But I thought this picture was kind of cool because it's really it's random. I really wanted to bring that in because there's not just one sperm cell that's racing to the egg. That's why they have that joke, "I'm the winning sperm." Okay, but here we go. We have the egg, and this is an electron microscope, and these sperm cells burrow themselves in, and once they're in there, that's when we get our zygote and our chromosomes starting to fuse together. So, do we need to do this naturally or in vivo? No, we have what's called in vitro fertilization. In vitro fertilization is kind of interesting. So, if you're having troubles conceiving, you can pay some money and they'll do it in a laboratory for you. And this is what that movie, we'll talk about Junior and Gattaca, were all about. Because those movies are all about in vitro fertilization. Okay, so what it is if we look here, we collect an egg and sperm cell from donors, whether it's themselves or a random donor that they pick from the list of people that are donors there. Well, we combine them in a little petri dish, and we manually, like this is a stand-up picture, we manually put the sperm into the egg. So this is our egg cell, and they would inject the sperm in here, and then let it grow, have a few different cells, and eventually we'd put those little embryos, okay, those little zygotes, after they've started to develop, into the womb of the uterus. Okay, so this could be expensive, so it's not cheap, and just to save your sperm or things like that, you have to pay money plus hormones and other things to maybe prepare your body. It can be kind of pricey, but in the end, if this is something you really want, this is an option for you. The babies come out normal. They're just babies. I mean, it's the same idea. They're made with love and science. That's what I like about this one. And that's why I thought these movies are interesting, because Junior is where they do a science experiment where Arnold Schwarzenegger does in vitro fertilization on himself. I don't know how that happens. I don't know how that works without the right parts. But so he's a dude carrying a baby through this process. And Gattaca is another cool one, because there's this huge futuristic society where having a baby normally is bad because you can leave everything up to science and pick the traits you want. So what would you want? Would you pick the traits if you could? And how would you treat somebody if you knew? So this is a really interesting film. Gattaca's great. So here's some of your baby photos of when you were a little zygote. Everyone kind of looked the same, but you can kind of see over time we have our nucleus fused together. We start to do this bubbling, right? We have cells and cells and cells. So the zygote does mitosis, and then we start to develop what we call the embryo. Okay, so once we start to really start to make all these different cells, we have an embryo. That's the second stage. And what's interesting about the embryo is you have two parts. This is where we split off. Okay, we got protozomes and deuterozomes. A protozome is a creature or an animal that starts to develop where we start to make the internal cavity first. So if you either make your mouth first, which are usually insects, worms, and invertebrates, you are called a protozome. If you make your butt first, you are going to be a deuterozome. Okay, so we're all a bunch of deuterozomes, which sounds bad, but really it's just the way we develop. And so eventually these inner layers too start to even get more specialized by turning into just your brain, skin, organs, and then this embryo turns into what's called the fetus. And you can see the development over time. We start to get more and more refined and more organs developed, so on and so on. So, what's the big deal with stem cells? You may have heard of it. So these embryos that we just talked about in the last slide, the embryos, or these developing babies, can be used for research purposes. So stem cells are undifferentiated cells. And since these embryos are not clearly defined as this is a brain cell, this is a skin cell yet, we can harvest these embryos and use the stem cells to turn them into basically anything. So there's like all these crazy cool possibilities. You could take stem cells and use them for testing different drugs. There's places like the U of M that have been using it to create a heart and other organ transplants, if you will. Treating different birth defects. Maybe we can use stem cells treating diseases like Alzheimer's and all these crazy diseases where maybe we're losing damage, even the idea of repairing nerve damage. We can maybe use stem cells that could get differentiated into those cells to repair them. So it's a really interesting idea and really cool application, but people don't like it because embryos, or developing babies, if you will, have a whole bunch of stem cells. And so there's this idea of, should we use them? Should we not? When does life really start? Does the pros outweigh the cons? I don't know. It's a really interesting topic, and I encourage you to research more about it and really develop your own opinion. But we're going to get into all of this, meiosis, babies. We're going to talk about all of that in our new unit of meiosis and genetics. So keep all of this info in your brain and ask questions along the way. So let's see if we how we did. Here is a quick matching section that I encourage you to do in your notebook of meiosis, gamete, embryo, in vitro fertilization, stem cell, and zygote, just a few of the terms you heard today. And then another mastery check, I put 2 in here. You could make a Venn diagram, which we started with comparing mitosis and meiosis, or a really good challenge would be to kind of summarize what you learned today in the video, but use the terms. Use what meiosis is and explain how it's different from mitosis. Explain what a gamete is and what happens to your gametes during fertilization, what happens after fertilization, and how we develop this fetus into a baby. So, lots of information there. Feel free to shoot me questions, and we'll see you next class. Thanks, everybody.